Hello, my name is Fredrik Zetterberg and I'm Director of Medicinal Chemistry at Galecto Biotech. This is a small virtual and global biotech company committed to the development of novel small molecule therapeutics directed at biological targets which are at the heart of fibrosis, inflammation and cancer. First of all, I want to thank the organizing committee for inviting me and making it possible to have a scientific meeting also in these strange times. Today, I will focus on the discovery of GB1211, the first orally available Galectin-3 inhibitor taken into man. All cell types are covered in glycans. If you look at the picture showing an artery, the beard-like structure you see is what we call glycocalyx. It is a layer of 10 to 100 nanometer thickness consisting of glycans sitting on glycoproteins, proteoglycans, etc. These glycans make up the glycocode, and there are more than 80 carbohydrate binding proteins interacting with it. One of these protein families is called galectins. It's a family of small peptides with size about 30 kilodaltons. It's a very old family of proteins, so it exists in all kinds of species from sponges, nematodes, and of course humans. There are a family of 15 proteins in mammals, where not all exist in human. They are soluble cytosolic proteins that pass in and out of cells via non-classical pathway. They are defined structurally by having carbohydrate regions and recognizing beta-D galactopyranosides. Galectins are divided into 3 different subclasses. The prototype consists of one single CRD that can be dimerized, the chimera, which consists of a CRD and a collagen tail, the tandem repeat that consists of 2 different CRDs and in the 1 N and C terminus. The galectins binds to N-glycans on glycoproteins to form lattices, and thereby they can control the surface residence time of protein on the cell surface. The galectins are also involved in the glycoprotein trafficking and endocytosis. So for example, by inhibiting galectin-3, one could inhibit the function of TGF-beta on myofibroblasts or CD98 on macrophages. There is a high unmet need for treatment of end-stage fibrosis. Fibrotic diseases occur in several organs, including lung and liver. The common feature is that fibrosis disrupts the organ's normal architecture and leads to organ dysfunction and failure. Fibrosis also makes some tumors, like for example pancreas cancer, resistant to treatment. 45% of the mortality in the Western world is caused by fibrotic diseases, and there are very few drug programs targeting fibrosis, despite this great need of new treatments. We have 3 compounds in clinical development targeting fibrotic disease. First off, GB0139, an inhaled galectin-3 inhibitor targeting idiopathic pulmonary fibrosis. Next compound is GB1211. With this compound, we're planning to target NASH in first round and then other fibrotic diseases. We also look into potential oncology application for this compound. In addition, we have GB2064, a LOXL2 covalent inhibitor targeting myelofibrosis. In the fibrotic process, galectin-3 is involved in mechanisms of 2 cell types. First, the macrophage, which differentiates into an activated stage where it releases galectin-3. In this way, it both self-amplifies macrophage differentiation and orchestrates the differentiation of the epithelial cells into myofibroblasts, which then deposits collagen, starting the fibrotic process. By blocking galectin-3, we thus have a strategy to block fibrosis by hitting more than one mechanism. Of the fibrotic diseases, IPF is the most rapidly progressing one. The patients have a median survival of 2 to 5 years. It's an orphan indication and 2 treatments are available. Unfortunately, these are hampered with side effects, and less than 50% of the patients in the Western world are on actual treatment. GB0139, or as previously called TD139, is the first compound to be developed by us. It's a safe, inhalable treatment with a strong safety profile and a confirmed target engagement. I will focus a little on this because these results are supporting that Galectin-3 has an effect on fibrosis in humans. In the clinical phase 2A trials for GB0139, we performed a lung lavage both prior to and after 2 weeks of daily inhalation. The compound was found to be safe and well tolerated. And also we could see that the plasma levels showed that the PK was dose correlated, and the lung lavage enabled us to measure drug concentrations and levels of, for example, Galectin-3 in the alveolar macrophages. These correlated well with the dose given, and not only that, we found that GB0139 had a consistent and dose-dependent effect on relevant fibrosis biomarkers such as PI1, PGF, BB, and YKL-40. This indicates that Galectin-3 inhibitor has effect on fibrosis in humans, and we are building on this data in our phase 2b trial. And since mechanism of fibrosis is similar in all organs, this gives us the confidence to go forward with the Galectin-3 inhibitor towards fibrosis in other organs. So, now we turn to finding of our oral Galectin-3 inhibitors and to the discovery of GB1211. NASH, or non-alcoholic steatohepatitis, is a spectrum of diseases starting from fatty liver disease that is abundant in 20 to 30% of US adults. 50 to 20% of these people develop NASH. These patients get steatosis, ballooning, and inflammation, and of course fibrosis. In the more advanced stages, NASH can turn into cirrhosis or hepatocellular carcinoma. Fibrosis is reversible from stage 2, and the more advanced diseased patients require a liver transplant. There are no approved products, so there is really a need for a treatment. There are many activities trying to find treatments targeting metabolic mechanisms, but our focus is of course complementary to this targeting the fibrotic side of NASH. When we started the medicinal chemistry program leading up to GB1211, the compound with the highest affinity was GB0139. This compound was first made by a PhD student in Ulf Nilsson's group at Lund University. This compound has high affinity and a very low oral bioavailability. We look in a radio-labeled biodistribution study in mice. They also suggest that this compound has a major biliary clearance. So to form a strategy going forward, we need to understand what drives affinity of these ligands to galectins and what limitations these structures have with respect to oral bioavailability. Can we use them as starting points at all? Let's start with affinity. Sugars in general bind in the millimolar to micromolar range, and for example lactose binds with 200 micromolar to Galectin-3. The CRD is polar and shallow. Galectin-3 and water is pre-organized to recognize beta-D-galactopyranosides with the sugar oxygens taking the place of water oxygens in the binding pocket. There is a low entropic gain upon replacement of this water. Sugar binding proteins often have an aromatic side chain interacting with the hydrogens of a sugar ring. In the case of galectins, tryptophan is a conserved feature interacting with the galactoside hydrogens. The hydrogens contributes to specificity of the sugars in addition to the water replacement. Therefore, a strategy has been to use beta-D-galactopyranoside for specificity and use unnatural substituents finding specific interactions to enhance the affinity. As already mentioned, we used GB0139 as a starting point for the oral program due to the unprecedented high affinity of this compound. These compounds are very, very polar due to having 6 hydroxy groups, and we had the idea of replacing or substitute these hydroxyls to decrease polarity and in that way increase the oral bioavailability. This turned out to be very difficult. One exercise we did was to start to methylate one hydroxyl at a time and starting with the ones that were not necessary for binding. Unfortunately, we didn't see uptake in CACO2 assay until we had methylated all the hydroxyls, and of course resulting in completely inactive compounds. We then turned to beta monogalactosides. They had a lower affinity and at best in the micromolar range. On the other hand, they were less polar and good uptake of CACO2 cells. We started to make libraries where we varied the aglycon on the right hand side with aromatics, sugar replacement, heterocycle, but never really got beyond the micromolar range of affinity. At one point, we left the reaction for too long and the more chemically stable alpha anomer was isolated. It turned out that the alpha galactosides had a tenfold improvement in affinity of its beta counterpart, and since these compounds are analogous and share similar physical chemical properties, we rapidly started to make different alpha analogues in a similar manner to the beta. To our surprise and joy, we found that compounds like GB1107 had the sought for combination of high affinity and low polarity, and therefore high uptake in CACO2 assay, and this resulted in high bioavailability in mice. The affinity of these compounds are highly dependent on specific and directional interactions such as pi, multipolar, calcium and halogen bonds. As can be seen from the X-ray of GB1107, arginine 144 is lifted up and form a pi stacking interaction with the phenyl group. In the induced pocket, multipolar interactions are observed between fluorines and the backbone. Both of these interactions are similar to how GB0149 interacts with Galectin-3. The real invention here is the calcium bond found from the sulfur to the tryptophan and the halogen bond from the chlorine to the glycine 182. The SAR of these compounds are almost digital. Removal of a chlorine results in the 3-digit nanomolar compounds. Replacement of the sulfurs with oxygen do the same thing. Removing fluorines completely kills the affinity towards Galectin-3. The affinity of these compounds is immensely improved over galactose, and so is specificity. So in addition to being orally available, these compounds are also the most selective Galectin-3 inhibitors published to date. Especially Galectin-1 has been difficult to avoid. We do hit Galectin-4 at 130 nanomolar, but this galectin is not so relevant systemically since it's solely expressed in the intestine. Our first thought was to select and go forward with GB1107 as our clinical candidate. Unfortunately, we found that it had a potential hERG liability from patch lab and guinea pig experiments. Therefore, we decided to go forward and optimize the structure further. We made a lot of variations on this series, and I will not bore you with all those in this lecture. I will focus on the optimization of a halogen bond, which I find is interesting and was crucial for the selection of our oral candidate. A halogen bond takes place between the lone pairs of a carbonyl and delta positive sigma hole in halides. These bonds have bond length between 1.5 to 2 angstrom. They are very directional, but if placed right, they can contribute to very, very high affinities. The sigma hole becomes larger when you go from fluorine to iodine, and in theory, this should enhance the binding if it wasn't for the potential stereo constraints of larger atoms. If you go from phenyl substituent and introduce a chlorine in the para position, we're placing the chlorine in the wrong direction, and the affinity is not much improved over the unsubstituted aryl. If we instead place the chlorine in the meta position, the affinity increases majorly. Switching to the bromine, we get a further increase, but with iodine, we lose some affinity. We have studied the series going from fluorine to iodine with ITC, and the thermodynamic contribution increases with the atom size, but when we introduce iodine, we also pay an entropic penalty, which results in a lower affinity. We can also increase the sigma hole by electron withdrawing groups or introduce nitrogens, for example, next to the halides. This doesn't result in high affinity improvement for a but we do get an improved lipophilic efficiency. High affinity compounds with reduced lipophility like GB1211 had no hERG liability. In addition, these compounds had higher free fraction and a lower hepatic clint, resulting in good dose rankings when predicting dose from hepatic clint. We therefore selected GB1211 as our clinical candidate going forward. This compound has a high affinity and has a good selectivity over the other galectins. It is clean in SUP, Panscrim, Mini-Ames and HERG. The solubility is surprisingly low for a sugar-containing compound and initially posed problems with respect to formulation. This was solved for our clinical trials. GB1211 has a medium hepatic clint and a free fraction of at about 4%. These two variables have the largest impact on the dose ranking between compounds. One thing that would have further helped us to gain confidence in selecting GB1211 as a clinical candidate would have been to have good in vitro tools predicting other clearance mechanisms than hepatic clearance, especially since we're dealing with sugar-containing structure and had observed biliary clearance without starting points. Anyhow, this didn't prevent us to get good exposure in man, as we soon will see. Remember that galectin-3 was involved with two different cell types important in fibrosis. IL-4 induces galectin-3 productions on macrophages. And in addition, galectin-3 can induce a self-amplifying mechanism by interactions with CD98, driving galectin-3 productions via PI3K activations. Both the thiazide like GB139 and alpha-galactoside like GB1211 and GB1107 inhibits this IL-4-induced galectin-3 release. In myofibroblast, we can inhibit TGF-beta-induced expression of collagen 1A2, alpha-SMA, TIMP1 and Gal-3 with the same set of inhibitors as for the myofibroblast. The IC50s of both of these cell assays are in the same ballpark as our KDs from a fluorescence polarization experiment. Human mouse and rat galectin-3 differs in positions important for binding of phenyltriazole ligands. Remember that arginine 144 was important for the pi stacking of this compound, as you can see from the X-ray. Below the aryl ring, alanine 146 is positioned. In mice, this position 146 is a much bulkier valine, potentially creating a stereoclash, and can be the explanation for a much lower affinity observed for this series of compounds in mice. In rat, arginine 144 is exchanged with the serine and the alanine in exchange for a threonine, and as a consequence results in an even lower affinity. This of course have consequences for selecting doses and testing these compounds in mouse and rat animal models. We've also made an arginine 144 to serine clone of human galectin-3, and we can see that we do lose some affinities. We don't have the pi stacking. We tested GB1211 and GB1107 in a carbon tetrachloride-induced fibrosis model in mouse. In this model, we induced fibrosis by dosing carbon tetrachloride IP for the whole study 8 weeks and start treating with compound at week 4. The PK profile suggests a once daily dose for GB1107, twice daily for GB1211. Both compounds reduced the fibrosis score significantly at 10 mg per kg, but not for GB1211 at 2 mg per kg. Both compounds also had an effect on alpha-smooth muscle actin. So we have shown that our galectin-3 inhibitors bind galectin-3, they have effect in macrophage and myofibroblast cell assays, and shown to reduce and protect against fibrosis in a mouse model. That in combination with having a profound effect on fibrotic biomarkers in IPF patients with GB139, we selected to go forward with GB1211 as our second galectin-3 inhibitor into clinic. GB1211 was taken through IND-enabling studies and into clinical studies. In the first SAD study, we found that the compound was dose correlated from 5 to 400 mg and had a clean safety profile. The crossover of PK curves indicates a change in the size of a tablet, which independently showed this dose correlation. We dosed up to 100 mg BID in the MAD studies, and GB1211 had a clean safety profile. This gave a good exposure in man with a 50-fold window over KD and a 3-fold window of 3 plasma levels over KD. Therefore, we have selected to go forward with 10 and 100 mg doses in the upcoming NASH study. We have discovered the first orally available Galectin-3 inhibitor and taken it to man. We have extensive clinical, preclinical and clinical evidence that Galectin-3 is a key driver in fibrosis, and we can inhibit it. Our lead compound GB1211 has passed clinical phase 1, and we now plan for starting our phase 2A study in NASH patients. Thanks to all my colleagues at Galecto Biotech and to my colleagues at Lund University. I also have to thank especially my colleagues at Red Glide and ChemPartner for excellent work in the discovery of GB1211. There are of course many other people who have been involved in this project, and I'm really grateful for all your efforts. You know who we are. And last, I want to thank you for listening and looking forward to take questions.